All right, so cytoskeleton, the next part, and very interesting. Some of the pictures that we talked about that, and there's a couple of things that you can see that when you fluorescently label different parts of the cell, and all these pictures were basically taken in separate shots, and they were stocked upon each other. So you can see the all the different cytoskeletons. These are the chromatids during the metaphase, which are aligned, and then you have the microtubules and the actin and everything in there. You can see that these are the finger-like projections, which are the villi or the villus singular villi, and this black part is actually the lumen of an intestine right here. This is the lumen of the intestine, and these finger-like projections, small intestine rather. This is usually the jejunum or the ileum part, which is the absorptive part of it. And what you are looking in here, that these finger-like projections, first of all, increase the surface area of absorption. And this red-lined structure that you are seeing, these are lined by the epithelial cells. These are lined by all epithelial cells, and in between line, they are not connected. There's some deep crypts in here, and although this looks like they are fused, they're not. There is deep crypts in between. So the food material or the digested material can get in, and this is an absorptive function. And the red things are what we call as a brush border. These are the microvilli projection of these intestinal epithelial cells. And those microvilli can increase the surface area of absorption almost like 600-fold, these tiny finger-like projections. And that is all over from this crypt to all over those and tiny cells. And the blue parts are the nucleus of each of these cells. And the green part is actually a protein called lamin that is on the basal side of it. So the red part is the apical side of those epithelial cells. The blue is the nucleus, and the lamin green is the basal part of it. So you can see that these are all polar cells in there, and shows amazing part. And at the bottom, that what you are seeing are the blood vessels and the lymphatic vessels. So anything like a carbohydrate, sugars, as well as proteins, which are water soluble, absorbed, they are transported into the blood. Whereas the fatty acid, cholesterol, lipid, essentially breakdown product of the lipid, they actually go into the lymphatics, or the lymph vessel actually take them to the liver, where they are processed, put into the LDL, and associated with your ApoB protein, and then delivered to each cell via blood. So just an idea that lamin is also intermediate filament, and these red parts do have these actins, which actually play a huge role in the cytoskeleton, of course, play a huge role in maintaining those microvillus projection as well. We will see in a second. All right, so there are some of the slides which are very self-explanatory, and if they're very super self-explanatory, I will just not spend much time in there. So you can easily see that this is like very straightforward in there. So you can see that these are some of the other nicer picture. So here, this is basically what we are seeing on the left-hand side is a electron micrograph, and this is a schematic of, think about that each of these cells that you are looking here is representative of one of those cells. So this red part is the microvilli, and those red things are the actins, which actually are maintaining those structures, and they can move as well, because these actin are in a dynamic state. They can break down, they can build those. And then on the basolateral side, you have other filaments like intermediate filaments, microtubule. There are some of the filaments which inside the nuclear membrane, nuclear lamina, underneath, there are some intermediate filaments which actually maintain the shape. And you can see that these are all over the place. You have these blue, red, and the things. They are basically representing the three different types of microtubule. You have the microfilaments, you have the intermediate filaments, and you have the microtubules. So these looks like to a cell which are sessile, means that they are fixed in one place. And you can see that there is a basement membrane, extracellular matrix, and they are not going anywhere. Whereas this is a representative of a typical cell which actually move. And the only type of cell, pretty much that the most common ones, are usually the white blood cells. Like red blood cells move by the flow of blood, but they do not have the ability to move by themselves. Whereas the most of the white blood cells, hugely important in immune function, they can crawl in and out of the artery that we are going to learn later part into the tissue space. And there is a huge amount of remodeling of these microfilaments to do those things. So basically, this is basically what we call as a filopodia. And these are the projections by which they actually do move. And even the pseudopodia that you have heard about in the amoeba, those are basically moving structure that they are moving. And this is basically building actins and and then dissociating them and rebuilding those in various conditions as well. So these are essentially an epithelial, a static cell, and a migrating cell. So we already explained that, what we are looking at in here. So these are some of the general considerations that are just very straightforward. So the actin we call this as microfilaments. These are helical flexible, and they are made up of these globular actins. And these are globular actin monomers. And here you are seeing the molecular structure, and you can see that they actually make a helical structure where you have these two of these are actually kind of twisted together. And these filaments are always directional. There is a minus end and there is a plus end of that as well. So there are two parallel protofilaments wound around each other. So remember that there are two parallel protofilaments. So there are two actin filaments. So one of the basic concepts, when the actins are in the soluble singular, like a monomer stage right here, we call this as a globular actin or a G-actin. When they are polymerized into this filament, which we call as a microfilaments, we call them as an F-actin, filamentous actin. So these are some of the general names and the considerations. Yeah, so these are the different microfilaments. You can see that where they are, different distribution of those. So just a couple of things that what we have, microvilli, we talked about that. This basically cell cortex underneath the plasma membrane maintain the cell shape. Adherence belts, we are going to learn that in the very last chapter of the course, which actually kind of attached the next cells to each other, glue-like structures, and we are going to know that those adherence belt. Filopodia, we talked about that. Lamellipodia, just two different names. Filopodias are just like a sharper structure. Thinner filopodia is a leading edge, which is a little bit blunt. Cell cortex of a moving cell. This is a cell cortex of a static cell. Stress fibers are usually long elements that extend throughout the bodies of the cells, which are motile. So this actually ensures that as the cell is moving forward, the cell don't tear apart. So those are called the stress fibers. And the contractile rings are actually formed at the very late end of the cell division, which is a cytokinesis. And after cytokinesis, cells are separated by these contractile ring. In a plant cell, it happens in a different way by a cell plate formation. But here, a contractile ring actually, and it actually what is called a cleavage furrow. And this cleavage furrow is basically invagination of these membranes, and they basically will fuse together, separating the two cells. So all these are done by the microfilaments, which are actin-based. So the second one, just a broad idea. So this is your intermediate filament, say that they are 10 nanometer, whereas these are about 7 to 9 nanometer. But the intermediate filament proteins, the main thing that you actually take home from here, that you know that the microfilaments are made up of this single protein, which is the globular actin monomer. This is the only protein that makes it. Whereas the intermediate filament is made up of many different proteins depending upon the tissues and the cell. There is no specific protein that makes it. Again, what we are going to see, the microtubules are also made up of the tubulin. There are alpha, beta, gamma, different types of tubulin, but they are specific proteins, whereas the intermediate filament proteins are most diversified. And we're going to see that. So here, what we are looking at, the intermediate filaments can be formed in the tight junctions, which keep the cells together. And you already know that tight junctions doesn't allow molecules to move from the basolateral surface to the apical surface. And you already know the function. So here, what you are looking at, that underneath the nuclear membrane, there is a nuclear lamina. And that nuclear lamina is one of the place where you have this intermediate filaments provide the structural strength that is required. Then we are looking at the microtubule. There are tubulin. There are several one of them. And this is actually a hollow tube-like structure. And this is the largest out of all. You can see 25 nanometer in diameter. And the tubulin monomers, there are two of them, alpha and beta. They are just joined head to head. So they have a plus and a minus end. And usually they do have an area from which they actually spread out all throughout the cell. And we call this, very important, repeatedly underline that, is the microtubule organizing center, or MTOC, which is known as a centrosome. And this centrosomes will duplicate, and then they will migrate towards, in a dividing cell, they will migrate towards the two different poles during the cell division that in the cell cycle we are going to learn about more. So you will see that this whole chapter will, again, we will have a lots of recap when we go into the cell division. All right, so these are some of the basic consideration of that. And here you have the distribution of three types of protein filaments. And what we mean by the similar distribution is, if you look at the microfilament, you can see that they are running in all direction of the cell. Whereas if you look at the distribution of the intermediate filaments of the microtubule, they are kind of radiating from the center of the cell towards the periphery. So we can say to some extent, again, this is, of course, you can, it's like how this looks similar, they looks very different. Of course, they look different, but the patterns are that they are kind of a radial from the center of the cell, whereas you can see that there is no radial arrangement. They are running in all different directions all throughout the cell. And that's basically is the reason that we call that the book, they have a similar distribution. So what we can actually ask from these questions, from these initial questions, that what are the similarities of the microtubules and microfilaments? Sometimes we ask is like microtubules and microfilaments are similar in which of the following? And say, for an example, they assemble, disassemble what way? Energy source they are using, we are going to learn that it will be ATP versus GTP, polymerization, depolymerization, how they differ. But one of the most important part that what you have, that the similarity between the microfilament and the microtubule, they both have a plus and a minus end. So that's one of the most important similarity. On the other hand, say microtubules actually assemble from the small number of location in the cell, which are the MTOCs, which is not true for the actin. So those are dissimilar. The only similarity that what you have, even the distributions are dissimilar, but the similarity only so far that what we found, the slides that we have seen, you have a plus and minus end, and we're going to talk about that. It's not here, but we're going to see that they also have a plus and minus end. That is the only thing that is similar. So monomer globular actin, polymer filamentous actin, or F-actin. So if you look at this structure, the plus end is called actually the barbed end, minus end is the pointed end. And what we mean by that structure, it will essentially look like this. This is basically the typical structure, that plus end is the barbed end. This is the plus end, and this is the pointed end, and this is the minus end. So they are stacked upon each other as well. So this is like a schematically think about this way, that what we mean by this barbed end, fast growing, and the pointed end. So let's take a look at the next part. So here we are looking at the actin polymerization step. So globular actin, they will polymerize by themselves. But what happens that if there are some seed or some of the nucleus where there are a couple of polymerized actin in there, the nucleation on top of this nucleus, the F-actin can be assembled much faster rate. So that's the process that what we are seeing. Now, there is a very interesting thing that actually happened there. So first, we have to understand that there are some structure where these filamentous actins are completely permanent and they do not associate or dissociate. So for the muscle cell, these actin filaments are super stabilized for the entire life of the muscle. They do not undergo those. But on the other hand, many other different types of cell, they actually undergo polymerization, depolymerization, and they most actively undergo these into the motile cells like your macrophages, neutrophil, basophil, all your white blood cells, which actually move, most dynamic nature. Now, there is an equilibrium of the globular actin and the filamentous actin in a cell, and there is a critical concentration for the G-actin. What we mean by the critical concentration, that if there is a cellular concentration of the G-actin at one point of time, and in physiological concentration, it's about 0.3 micromolar, approximately. Do not have to memorize the values, just saying 0.3 micromolar. So what happens if the values of this above that plus end and the minus end is going to happen, that they're going to be a polymerization, and if not, it's going to be depolymerization. So when a actin filament is basically neither growing nor shrinking, that means that there are plus end, you are just adding as much, and from the minus end, you are depolymerizing as much. We usually reach a steady state where it reaches this critical concentration. So what happens that when the G-actin, globular actin, is greater than the critical concentration, G-actin will polymerize into F-actin until the critical concentration is reached. Whereas if the globular actin is less than the critical concentration of the F-actin, will depolymerize into G-actin until that's what it is. Now, in an actin filament, the plus end critical concentration and the minus end critical concentration actually varies. So we have a couple of critical thinking questions regarding in that line, and we're going to explain that as well. We'll come back to that, the steady state, the critical concentration problems. So here is a typical question that we actually asked. We already talked that when there is a nucleation, we can have these things growing faster. So we can give you a graph like this, and we like to give you some options to predict that from here, you are just doing, say, for an example, an in vitro actin polymerization experiment using different conditions. And somehow you actually forgot to label the tubes, that where you have added just the G-actin and where you have added some of these nucleus in the test tube. But you just measured those and you plotted the graphs, and then you see that there are two types of graph that you got from these two experimental setup, without nucleus and with nucleus, with nuclei. In one set of graph, you got this sigmoid graph, whereas here you get this kind of saturation kinetics. So what it means that if you do not have any nucleus, you are going to have a slow start, essentially a lagging type of phase, and then you have an exponential growth. But if you have added the nuclei right from the beginning into the test tube, there is no lag. It will reach the exponential phase or elongation right there. So you can easily, from these two types of experiment, that in an in vitro experiment, whether you have added nucleus or not. So it's very straightforward, just the interpretation of these two graphs. This will be given as a picture, and you can tell that from there. All right. So here we are looking at the barbed end and the pointed end, that as I wrote in a little bit different ways. So the globular actin actually bind to ATP. And right here, which is the barbed end, is the plus end, and the pointed end is the minus end. So what happens that the ATPs, which are charged, the G-actin charged with the ATP, they will be added to the plus end. And there is a phenomenon which we call as a treadmilling, because if there are new ATP-bound G-actins are being added at this end, you are going to see that they are going to move towards the minus end. Now, the G-actins, this is the important part, they have an intrinsic or built-in ATPase activity. That means the G-actin in this filamentous actin, of course, two of them, they will hydrolyze the ATP into the ADP, adenosine diphosphate, and the inorganic phosphate will go up. And what happens that when the ATP-bound globular actin is much stable in the filamentous actin compared to the ADP-bound actin, when the actins are ADP-bound, they lose the affinity or the integrity to be kept into the filaments, and they tend to fall off. It needed to be aided, but the affinity actually varies. ATP bound, they are like to be staying in the filamentous form. ADP bounds do not. So as they treadmill towards the minus end, these intrinsic ATPase activities actually hydrolyze them. And that's the reason from the minus end, we see there are depolymerization from the minus end, whereas the polymerization, which is ATP bound globular actins, are added at the plus end. And we will go into a little bit more in detail about these things as well. So here is an experiment, and we can ask these questions. So one of the important things is that instead of an ATP, we can add a non-hydrolyzable analog of ATP, adenosine triphosphate. Now, this adenosine triphosphate, which is just like a nucleotide, just like the ATP energy currency, but remember that it's a nucleotide. So you can build a molecule which looks like an ATP, and it will bind to this pocket of the ATP binding pocket, but it is slightly different, and the ATPase function of the G-actin will not be able to hydrolyze it. That means that ATP analog, which is similar to ATP but not ATP, is not going to be broken down into the ADP. Once bound, it will be irreversibly bound to the G-actin. It cannot be hydrolyzed. In that case, what we see that the depolymerization is essentially blocked. That means that, think about that all of these are as if they are all ATP charged, but they are not ATP charged, but they are similar to ATP. Once they polymerize, they will stay in there because these analogs are not hydrolyzed into the adenosine diphosphate, which actually gives the idea that the role of an ATP hydrolyzed to ADP does cause the depolymerization. This is one of those. Now, what we are going to see that this is the natural depolymerization of the actin in the cell, but this is not fast enough. So we need some additional protein where we need an extremely fast severing of these actin molecules to speed up the process, particularly when the cells are moving. But this is natural. When the cells are gradually doing this, this is okay. But many a times we are going to see that when we need these remodeling of the actin need to happen extremely fast process, just this natural process of depolymerization and polymerization is not sufficient, and you need additional proteins that we're going to talk about. So here we are looking at the unequal growth rate at the two ends of actin, where the CC right here, critical concentration, is much, much greater in the CC at the plus end. So right here, you are looking at the minus end and see that critical concentration is much higher at this end compared to the critical concentration at this end. Due to that reason, when this happens, globular actin monomers, or the G-actin monomers, are preferentially added to this end, whereas from here, they get dissociated. And you can actually fluorescently label these globular actin molecule by recombinant DNA technology. You have a fusion protein, like you can just fuse them with a fluorescent proteins, and you can actually see them treadmilling. This process is known as treadmilling, that they are being added and then they are moving towards the minus end in this process as well. This can be actually done in an in vitro experiment. So we will look at some of these value and explain that what is going on in here. So this is one of the formula that definitely remember, and there is a critical thinking question based on that, and I will explain that. So what we are looking in these conditions, that if we just looking at the minus end, this is the plus end. So what is being shown in here, that this big arrow shows that at this end, the globular actin molecules are added at 12 micromolar per second. That is the rate of association. On the other hand, the rate of dissociation at the plus end is 1.4. This is the value. So if you look at the formula of the critical concentration, it is the rate of dissociation over association. So you can see that, remember that is 1.4 over 12. And the critical concentration is 0.2. So this, remember that this value 1.4 is right here, and association is below. So it's 1.4 over 12, and you will see that will be about 0.12 micromolar. This is the critical concentration at the plus end. And if you look at that, we already know from there that in general, the plus end critical concentration is much, much less than the critical concentration at the minus end. So let's see that minus end, what happens. In the minus end, rate of association is 1.3 micromolar per second, almost like tenfold less. So if this is 13 and 1.3, it will be exactly tenfold, but almost tenfold less association. But dissociation is not that much away. It's about 0.8. That means that if we just, like a normal condition, 1.3 subunits of G-actin are going to be added per second. 0.8 is going to be, again, it's an average, of course. That's why it is not a round number, like not 1 or 2 or something. So if you just calculate the critical concentration, it is the rate of dissociation over association. So 0.8 over 1.3, and you get 0.6 micromolar. See, that is 0.6 versus 0.12. Much, much less here, much, much higher here. Now, see that what is happening. I'm just giving you a condition, which is, again, cell-wise, it will vary, but just giving an example. Suppose you have a concentration in the cell about 0.3 micromolar. See, 0.3 micromolar. This is the physiological condition of a cell, which is very common, 0.3. See that the 0.3 micromolar is higher than this. This is higher than, this is higher than this. On the other hand, if you look at this right here, the critical concentration is actually higher than this. So this is basically goes like this right here. So see that this is the least, this is in the middle, this is the highest. So in a physiological condition, see that 0.3 micromolar, this end will add essentially and grow. On the other hand, see that this is higher the value than the 0.3. So there will be a net dissociation from that minus end. So this is the concept. And where do we get that from? So if we just go back, previous slide, see right here, we get that when the globular concentration is greater than the GC, G-actin will polymerize into F-actin until the critical concentration is reached. See, when the globular concentration of F-actin is less than this, it will depolymerize into the G-actin. Remember that what we are looking at, this value right here, this is the globular actin concentration. And see that this is higher than this value, but lower than this value. As this is higher than this, there will be an addition. As this is lower than this is, so remember that this 0.3 that what we wrote here, this is the concentration of the globular actin is right here, ATP G-actin in here. And this is the critical concentration at the plus end. This is the critical concentration at the minus end. Now, there was a critical thinking question. Suppose you have a value, I'm just saying that right here, you have a 0.5 micromolar. This is a value like this, 0.5 micromolar. So 0.5 micromolar, suppose I'm just taking random arbitrary. And then you see that at one end, suppose this is the plus end, and here the value, you have the minus end. There is two different values. And what you see that there are two values, both of them are actually less than this. So say for an example, suppose you see that there is a 0.4 right here. This is actually 0.4. And 0.4 is indeed less than this. On the other hand, suppose you have an arbitrary value right here. You have a 0.1 at the plus end. This is much less than the 0.5 compared to 0.4. This is far less than this. So that is what the point is. Now remember that in this condition also, we will see dissociation from that end and the association from that end. Because remember that here, it is ATP actin. When they are treadmilling, the ATPs are dissociated with the ADP. So even in the condition of this critical concentration, the ADP actins do have less affinity and they will gradually dissociate. So what will happen that if there is a concentration where you see a steady state or polymerization, always look at the value, and this is a typical example, the value which is far smaller than the physiological concentration, that end will get the addition. Which is closer to the critical concentration will get the dissociation. And again, this was an example in the critical thinking question. But usually what you will see that if we just give you this example in the exam, so suppose I will just say that if you see a 5 micromolar, not even 5 micromolar, if you see 1 micromolar one end, that is much less. If you see 10 micromolar one end, that is greater than those, and you will be able to understand that. So 10 micromolar, this is 5 micromolar is a G actin concentration. And if I say that 10 micromolar at the minus end, of course, it is larger than this. 1 micromolar at the plus end, this is definitely less than 5 micromolar. You will have association at that end. But this is the concept that if both of them are actually less than the globular actin concentration, always see that which is far less compared to the other. That end will actually grow comparatively due to the ADP actin hydrolysis. That's basically the concept or the explanation of the critical thinking question that you have. All right, just a couple of examples of these metabolites. These are all toxins, very, very toxic material. And these are widely used in the laboratory as well as in research. These are cytochalasin. There are many types, A, D. These are fungal metabolites, and they cause this depolymerization of this actin. So essentially, these are absolutely cytotoxic and cause these problems. Then latrine this is from a poisonous sponge, the marine sponge, and that was also called depolymerization. On the other hand, see that what phalloidin is doing, and that's why definitely remember, this is a natural alkaloid from the Amanita phalloides. This is the death cap mushroom. And if you are in South Carolina, in the Georgia area, these are all over the place. I mean, like they look like these general white mushrooms, but they're, if you just take carefully them out of the ground, their bottom part of the stalk actually is it's like an egg shaped. It's bulbous a little bit. Look at some of the pictures in the internet, and you can see those. Absolutely dangerous, because what they do that they stabilize the actin. So the cell will completely lose dynamic nature of that. If someone actually consumed these mushrooms, it is hepatotoxic, and usually the symptoms occur after 48 hours or 72 hours. And if they are alive and tell you the story, probably they need liver transplant or those kind of things. I mean, like in a good amount of poisonous dose, this is almost always deadly. So, but very interestingly, just saying is like we have a cell biology laboratory for the undergraduate, graduate in-campus classes, and they do some of these experiments with the cytochalasin and the phalloidin and see their stabilization and the effect on some of the cells. These are very, very commonly used cell biology material. Just remember that the mechanisms are different with those. So you will have this slide in front. So we'll just ask that what it does. Just remember that cytochalasin that will basically depolymerize, where the phalloidin essentially stabilize the basic difference. So basically what happens, so these are all good. As you can see that it just shows that how this is like all over the place. You can see leading edge of the cell. Now you can see that this leading edge is empty, because this is cytochalasin is basically destroying the microtubule. And when you do the immunofluorescence, see that this area where you had all the leading edge and the microfilaments, they're gone from there. So that's basically what it shows. All right. So after that, we are looking at these actin binding proteins. As we said, that we have these polymerization, and these polymerization happens when it's ATP charged. Depolymerization happens when it is ADP. ATP is hydrolyzed to ADP, but that is sometimes not sufficient. Some of the dynamic cells need things to happen in a much faster rate, and some of these proteins play a role in here. So definitely, this is an important slide. So that's why it is marked. So know all of these proteins. Somewhere we will ask questions about these, but let's take a look at that what we are looking at in here. So all of them are important. We will just ask the function of that. This is the plus end of a filamentous actin, minus end of the filamentous actin. So the first one that what we have is this cophilin, and which is not shown. There is also the protein, which is gelsolin, which will also do the same thing. So cophilin and the gelsolin, both of them are basically called the severing protein. We already know that at the minus end, the depolymerization naturally happens when the actin the actin ATP is hydrolyzed to actin ADP. But the cophilin as well as the gelsolin, they bind to these structure and sever them in chunks. So this happens in an extremely rapid millisecond time when the cells need to change the dynamics. So the structure. So severing protein, very rapid depolymerization from the minus end, covalent and the gel solid. So what we are now looking here, this D means that this is ADP bound globular actin coming out, and covalent is dissociated and it can bind again. So this is the ADP bound, which is what we called as an uncharged globular actin in the cell. So this actually binds to a protein which is called the profilin right here. So the profilin changes the conformation of the ADP-bound actin to some extent so that it now can exchange the ADP with the ATP. Profilin helps the ADP to move out and replace it with an ATP. So this is basically a positive regulator in that sense. So profilin is a positive regulator because the ADP-bound actin cannot be added to the plus end. It must be replaced by the ATP, and profilin actually helps in that process. So that's why it's a positive regulator. Now, the next part is the most important part. If the cell is requiring ATP actin to be added, and it's in a state where it is constantly needing these polymerization of these actin, these ATP actin with the profilin will be added to the plus end. Profilin will go out, recycle back, and this is addition. But the cell doesn't need to make these actins if it is in a resting state or something like that. So at that time, how you prevent the growth of these filaments? So the cell decide at this time, I do not need to grow. I'm in a steady state. I do not need to grow these actin filaments. Whatever their lengths are, I'm going to keep them just like that. So at that time, the treadmilling and this process will happen, and gradually some actin will be added, some will be taken off at a much slower pace. But there is always a very large pool of globular actin in the cell that will remain. Now, the problem is that if all these ATP pools are associated with an ATP and they are all with the profilin, they will tend to be added in here, because they will increase. Remember that these ATP-bound actins, they are going to increase the concentration of the cellular concentration of the globular actin, and they will basically try to be added on the plus end. So how you prevent that? So in that case, this thymosin beta 4 comes in, which is a negative regulator. And why is a negative regulator? This is a sequestering protein for the ATP bound actin. So what happens that when the cell decide that I do not need to add ATP actin and I do not need growth of these in a very rapid manner, thymosin beta 4 will bind to the ATP actin. And at that time, the ATP bound actin with the globular actin with the thymosin beta 4, these are not available to be added onto the pool of the ATP. So these cannot be. So this is basically sequestering or hiding the ATPs from the globular actin pool of the cell. When thymosin beta 4 binds, they cannot be added to the plus end. Now what happens when the cell actually gets a signal that I need to grow these, thymosin beta 4 will release the ATP actin and they can be added. So thymosin beta 4 goes in that. So they will just release them in the pool of the ATP actin, and very, very rapid process, they can be added to the pool. So that's essentially that the covalent and gelsolin, these are the severing protein, depolymerization. Profilin is the protein which helps exchange the ADP with ATP and addition to the plus end. And the thymosin beta 4 regulates the amount of globular actin available to be added to the plus end depending upon the condition. And as it is a sequestering protein, it is a negative regulator of the actin. A typical type of question is that in an in vitro experiment, addition of which protein will cause a depolymerization of the actin. And remember that those will be your covalent and gelsolin. If you're just doing an in vitro experiment, if you add those, so very straightforward question. So after that, we are looking at these actin polymerization in the motility. and know the steps, which is the step 0, where the trailing edge must adhere to the substrate before a lamellipodium can be extended, filopodia or lamellipodium. If it is blunt, lamellipodia. If it is finer, it's filopodia, just two different terms depending upon the dimension. So you have an extension adhesion, translocation, and the deadhesion, and these are all driven by the actin. And the only thing that, in addition, that we are going to see that the interaction between the matrix and these leading edge, we are going to learn that in the cell junction. So here you can see that there is a focal adhesion right here. And here you have direction movement, and this is being extended, and you have a new adhesion. And then you have a translocation of the cell body movement, and then you have an old deadhesion. Very straightforward. What are the different steps? 1, 2, 3, 4, 5. You will have these. So here you are looking at the different, so profilin, remember that, that's a positive regulator. ATP actins are all bound with that, so they are found in these leading edge. And these are the zone of polymerization. And you can easily see that these are all cross-linked polymer of actin at the leading edge. And now we are going to see that how these cross-links essentially happen. So there is a protein called this ARP23 complex. It's a large protein with 7 subunits, and just only 2 of them actually shown, which are ADP bound, 2 and 3. These are responsible for the branching, extensive of these branching that can only happen with this ARP23 protein. So how this happens, this is a very interesting way that what happens. There is a nucleation promoting factor, or NPF. And you can see that WCA is essentially the 3 domains, the 3 different domains of this nucleation factor. So you have 2 of these nucleation factor. On these 2 nucleation factor, the 2 actin subunits associate. In absence of NPF, ARP23 will not be recruited. So what happens that once you have this actin-bound NPF nucleation factor, then the ARP23, which is this, is recruited in the middle. So very carefully see that. This is your nucleation promoting factor, step 1. Then you have the actin added on top of that. Then you have nucleation promoting factor, actin, nucleation promoting factor, actin, and in the middle, you have the ARP23 complex. This is the complex which can associate with the actin on the side, not at the plus end, not at the minus end, but it can associate on the side. And from that end, you can grow another molecule, which is basically the branching process. So just remember that in the absence of NPF, this ARP23 will not be recruited. Branching of actin will not occur, which is essential at the leading edge of a motile cell. The motility of the cell will essentially cease. So sometimes we can just ask you some of these questions that you have a mutation where a cell cannot produce the nucleation promoting factor, or there is a mutation where the cell cannot produce an ARP23 complex, and this is the motile cell. What can happen if you do this, or if you just have an inhibitor of NPF or an inhibitor of ARP23? And if we just tell you that it's a motile cell, you will understand that this branching will not occur, which is essential for a motile cell, and any movement will essentially cease in that process. So this type of question that we can ask. So these are just mediated branching that you can see. All right. So here also, what is the overview of that? A couple of different things. So you have the plasma membrane, and the arrow actually shows the direction of the move. So what happens that the actins actually push the plasma membrane in this direction, and the plasma membrane stretch and forms a sharp filopodia. And if it's broad, then it forms a lamellipodia. So what you are seeing, that if you take a look at this is a plus end right here. This is an ARP23, and this is a plus end. And what it shows that you have profilin, and this is ATP G-actin. They are being added to the barbed end. The white parts are showing that they are now hydrolyzed into the ADP. And in this end, you can see that cofilin and the gelsolin, these are coated those ADP actins. So from this end, it's going to get minus end. Remember that the ADP G-actins are going to get recycled. So what happens that if the cell is going in this direction, you will have addition on this end. So suppose you have now a critical thinking type of question here. So let's take a look at that the cell was going in this direction initially, in this direction right here. So which is this. And then it decides that it does want to go to a different direction. It now wants to go in this direction instead of going in here. So what will essentially happen? If you carefully look at that, there are some capping proteins in here. There are some capping proteins. And the filament is basically a cross-linking protein. So what happens that in order to change the direction from this red side way to this green way, all these actins, what you have, they add these capping proteins onto the plus end. And that will block the polymerization of the actin on the plus end. There will be no polymerization on the plus end. But the capping proteins will not cap the actins, which are, I'm just drawing the other ones as well, other ones which are facing in this direction. These are all plus end of different actins. They will not cap that. So essentially all the ATP actin, ATP G actin, they are now polymerized in this direction. So everything will grow towards this end, but nothing will grow in this end because the growth in this red direction will be stopped by these capping proteins. So in this way, very, very rapidly, and this actually happens in literally millisecond time because sometimes these neutrophils chase these bacteria to, you can just look at some of the YouTube videos, there are supplementary videos, and they need to do this change in direction in a very, very rapid manner. And this can actually happen in a very, very rapid manner that they will basically cap everything and build everything in there. From there, they will build everything in there. So, it is really, the process is extremely fast, and sometimes it's beyond our comprehension that at the molecular level, that how fast these things can actually go. So know the function of each of these proteins. They're all in front of you. There's no big deal. And here, very interestingly, what happens, the Listeria monocytogenes, this is an amazing bacteria, and it usually, once it's infected, it doesn't go out of the cell, and it basically hijacked the actin polymerization of the cell to move in between the cell. So once it gets into the cell, it actually propels on these actin tails. And then what can happen that they basically can propel on themselves and they can protrude themselves with the plasma membrane. So they just come in an extreme force onto the plasma membrane, and the plasma membrane actually tears up, and then you can see that it is basically associated with the next cell. And this is how from one cell to the other cell, these bacteria can move. And what is the advantage of that? If they are inside the cell and not in the interstitial space, your immune system cannot understand that you have this infection, essentially escape from the immune system. So here you can see the bacteria and the actin polymerization tail, like the comet tail-like structure, that how it does that. So how Listeria does this? So there is a protein associated with that is known as ACT-A. And the ACT-A is a protein which is active polymerization of these protein can actually happen. So basically just think about that. And these are your ARP23, and you see branching, and these are the adding. So basically what happens behind ACT-A2, just think about that as if this actin is a staircase, and it is building that staircase and climbing on top of that. So here the staircase is dissociated. All these free actins are added onto the staircase, and they are just climbing on that staircase just to move forward, just giving an idea. So this is actually an extremely fast movement, and you can see the supplementary video that how fast they can move in there. So these are some of the very interesting pictures that you can see. All right. After that, we are looking at a particular slide. Should have a mark in there. So definitely know this GP1B9 protein. So definitely remember this one. So what is it showing? Platelet. And if there is a clot, here is a cell, and it can change its dynamic nature. And you can see that this cell is flattening out, and it can spread itself like this. And this can form below a clot, And this is a protein which is called a GP1B9. This is essentially a transmembrane protein. And this cross-link with the help of a filament, the actin filaments, to build this. So filament, we already saw that beforehand. Filament is a cross-linking protein. So where we go in here, cross-linking protein, filament, and this is what it is doing, that GP1B9 transmembrane protein interacting with the clot. And underneath the clot, this is basically spreading out to block that. And the filament interacts with the actin filament. So definitely, so filament is the one which cross-links. So cross-linking protein, underline that filament. Another protein that was important in there that we talked about is the CAPZ. That's a capping protein. And this CAPZ caps the plus end of this. You can see the plus end. And we are going to see that there are other protein like tropomodulin that we are going to see that when we go to the muscle cell. We'll talk about the myosin, tropomodulin, nebulin. These are the ones that we are going to talk about that when we go to the muscle cell. And here, what the idea is, we do not need to ask any question in here because this will come all together when we do the signal transduction. So these are the signals from the soluble factors, other cell extracellular matrix. And the signal transduction pathway essentially remodels the cytoskeleton, which organizes the movement of the organelles, cell shape, movement, contraction. And this is the process that all these things, actin polymerization, is actually driven by the signal transduction. It is just not random. When the cell actually receives a signal, it will make these changes. All right, so let's go to the part 2. And we are looking at some of the actin binding protein. We already talked about that. So the ones which are important here, we are able to learn about the myosin, capsid tropomodulin, titin, nebulin, and troponin tropomyosin. These are the ones which we're talking about. So the first thing is that actin-based motor protein, which is myosin. So typically a club-like structure. There are many different types. Club-like structure where you have a rod or tail domain, then you have a neck, and then you have these golf club-shaped head region. And the neck domain is very important, many functions, and these does have hinge which can flex depending upon whether they are binding ATP or not. So this is a myosin 2, just a typical structure. This is the most abundant myosin and the muscle myosin, but there are more than 20 different types of myosin. They all vary. So this is your typical myosin 1. You can see these are some of the calmodulin light chains, myosin 2, which is a dimer, regulatory domain light chain, myosin 5, almost similar, but still small difference. But the important thing that you need to remember from here, just remember that all of those, myosin 2 is the most common. That's one of the things. Most abundant myosin is the muscle myosin, which is myosin 2. But myosin 6 also underline that this is the only myosin that actually goes from the plus end to the minus end of the actin. All other myosins actually move towards the plus end of the actin, not the minus end, except the myosin 6, which has a different movement. It moves to the opposite direction. Rest are all plus end directed motors. That means they all move to the plus end of the actin. All right, so here, know this very well. And the thing is that as the material is in front of you, this is very, very easy. So here we are looking at the muscle contraction cycle, and we call this as a cross-bridge cycle. That is actually the word that we use for that, cross-bridge cycle. What a cross-bridge cycle is you have this myosin head right here, and you have the actin minus end, and this is a plus end. There is a pocket, which is an ATP binding pocket, and it's not bound. In that condition, the myosin is actually bound to an actin molecule very, very tightly at this point. So when ATP comes in, ATP binds to that pocket, and that is the trigger. So the head actually dissociates from that, and then it is hydrolyzed. The ATP is hydrolyzed to ADP and the PI, and both of them are still bound. And in this, the head actually pivots and binds to a new actin subunit. And then the PI is released, the phosphate is released. At that time, the pivot or the power stroke happens. So this is the one that is important. So remember that when exactly the power stroke happens, a lot of people say it's like when the hydrolysis of ATP happens, the power stroke. It is actually not that. the power stroke happens that when the inorganic phosphate actually leaves, that is where the power stroke actually happens in that process as well. And see that ADP is still bound. And once the ADP is released, it goes into the rigor state. So if you just remember that where the word the rigor mortis or the rigor mortis comes in, that what settles out after the death. So the ATPs depletes after death, and they are all hydrolyzed. There is no available ATP after 2, 3, 4 hours after the death. And all these muscle myosins are bound to the actin and in this rigor state. They cannot be separated out. And that's what causes the stiffness. So that's your rigor mortis. So calcium is required. And these are some of the things. Myosin 6 moves. Just remember those as well. Very straightforward. We just ask you what happens when power stroke happens, when is the rigor state happens, when the head dissociates from the actin, when the ATP binds. So these are the type of questions that each step, and this will be available to you. Just make sure that you familiarize yourself with those. All right. So these are some of the experimental consideration, and there are some questions that we actually asked. You will see that in your critical thinking question. So scientists were trying to determine that what are the different things that can modulate. This is a myosin head, and the neck region is the most important part in the cell. And what happens that if they add more amino acid, that means the protein length is increased in the neck region, their velocity actually becomes faster. And the reason for that, because their step size actually increases. So here, this is the wild type. So that means that there is nothing added in here. This is the original wild type myosin. Here, suppose you made a recombinant myosin with 1 amino acid, 2 amino acid, and 3 amino acid. So if this was the basal rate, then see that in each step that you are adding 1 amino acid, theoretically, the step size increases and the myosin essentially speeds up. So this is how the myosin 5 steps. This is a hand-over-hand step. And now remember that this myosin 5 movement is actually a little bit different than the myosin 2 movement. This is your myosin 2, which is the muscle myosin that what we are seeing. Whereas these myosin 5, these are associated with cargos. If we just take a look at, you have a cargo right here, and this moves these cargos, vesicles, and other stuff from one part of the cell to the other part of the cell. And the scientists actually found out that their step size, you can see that this is the neck region. They did a lot of these experiments in here, and we have one of these examples with some data from there. And you have this alternating leading heads. That means that this steps first, then this one will step here. And this is your alternating leading head. And the permanent leading head is basically scooting. So this comes in here, and then other one will come in here. And then this will be always, so this you scoot right here, and then you move this one to the other. So this will be always the leading head, and it will scoot the back foot back. So this is basically 36 nanometer versus 72 nanometers. And these are some of the ways that they can predict that what kind of movement is associated with that. And the alternating leading head is basically hand over hand walking movement that's in there. There's some amazing with the microtubule based, particularly dynein, kinesin. You can see these supplementary videos, some of the movies of Ron Vale who discovered those in your supplementary area. Those are some extra material, but again, not needed for your exam. But if you're just interested in that, you can just take. So here, typical experiment, what it shows, this is your time and this is your position. So this is what you're looking at is their speed. These are the steps. So basically what is happening in here with the time, these steps are basically oscillating and then they are stepping out. So this is the step size, what you are seeing. This is the position. And see that all of them are not exactly the same. And Kelly actually asked a question that we have this picture where we had like a 73.75 nanometer was the average step size. So how you do this? So remember that you are doing this experiment in an in vitro or a single cell, whereas you have a single molecule labeled with a fluorophore, and you are measuring the movement of that single molecule in a super high resolution microscope. Extremely sophisticated setup. Very few labs in the whole world can actually do these kind of single molecule super resolution microscopy. And they are getting the data that in between the initial and the final, how much distance it is. And you see that it's nanometer. It is a single protein molecule on these single molecules. So I mean, like when you are measuring a fluorescence, which is, remember that you cannot do electron microscopy. Electron microscopy, everything is frozen in time. There is no dynamic nature in electron microscopy. This is light microscopy, super resolution light microscopy, where you are detecting these molecules in a dynamic nature, and it's in the nanometer range. So there will be a little bit of difference in the data collection, and that's why you have to take that average. Here, when it is bound, there is no ATP associated with that. And now an ATP comes and bind in here, it dissociate, and it will just do a Brownian motion, and it will just try to move forward and do this Brownian motion constantly. And then suddenly it will be able to step. And this is happening in microsecond time, but still you will have these rapid Brownian motion because there are water molecules all over and the protein is interacting with the water molecules. So they will just randomly oscillate and step. And sometimes it may step on this one. Sometimes it may step in this one. Sometimes it may step this one. So basically when we are walking as well, our each and every step may not be the same. So if this step just the glomerular actin here, here, and here, their step size would be different. And that is in reality what is essentially measured. And then they have the average step size that is measured. So a typical question that we actually asked, and I'm just again addressing that you have a myosin, which you have a recombinant myosin that you made by addition of 3,4 amino acid in the neck region. And its step size is essentially this. Say, for example, it was like a 73 nanometer or something. And then we just give you a couple of examples in which direction it is moving, whether it was moving towards the plus end or the minus end. Remember that whenever we have added anything in the neck, that means the step size is increased. So the recombinant that we are measuring is this. This is the step size. So the wild type is the original from where we made the recombinant. So the first thing is that first take a look at the wild type step size value. That should be always less than that. So if this value is 73, the wild type should be anything less than that, 72, 71. Never could be 73, or if you see 73 or 75 or 76, immediately that cannot be rejected because you have less amount of amino acid in its neck compared to this. So the value should be less. That's number one. And the second thing that what happened is that the example was there with myosin 6. This is the only thing that moves towards the minus end. Rest all are plus end. So it will move towards the minus end. So that was basically the question. So take a look at that and you will see that. All right. So just two different types of things you can see: membrane association, endocytosis, contraction, organelle transport. As I said in here, they're different ones. And these are just straight examples. Now we are looking at these myosin 2 molecules, which are aggregate to form these thick filaments. And these are found in the middle portion of the muscle cell. So let's take a look at these muscle cells. If you just look at a myofibril, this is a single muscle cell and it's multinucleated. They are formed from the myoblasts, which are single cells, and they fuse together laterally and they are multinucleated. So you're looking at a single muscle cell. Now, if you take a single muscle cell, then this is a typical structure of the entire muscle cell. And they are organized into these myofibrils. So these are these rod-shaped myofibrils going to the entire direction of it. If you now take a myofibril, you get this structure, and end to end, they are arranged into contractile units, which we called as a sarcomere. So just think about that. This is a flat part of a coin, a rod, again, a flat part of a coin. This is one contractile unit. Another one starts here, right here. Another sarcomere starts here. So end to end, it could be hundreds and thousands of sarcomere, depending upon the length that they are connected end to end. This is completely continuous, and there is nothing that they are separate. This is just how we study it, because one sarcomere, the things that happens in one sarcomere, that will happen to the next one, that will happen to the next one. All these things are simultaneously happening, but it's easy to just read just one sarcomere to understand the entire muscle physiology. So if we look at this, that just correlate this picture from that right here, this one is the Z disc. And this is basically a flat, round, coin-shaped structure, because see that this is round. And that is essentially this line. This is a Z disc. So remember that this is two-dimensional, but if I just really have to draw this like this, and I am just looking at this part straight from this side, that's why we're seeing this. So this is a straight line. But remember that this is round like this. So this is one Z disc to one Z disc, and this is one sarcomere, which is one contractile unit. So now here, what are the things, how they are associated? So the actin filaments are stacked up like this, where you can see that these are the barbed ends. So the barbed ends or the plus ends are associated with the Z disc, and the minus end, which are the pointed ends, are towards the center of the sarcomere. The same way on the other Z disc. So you can see that the actins are associated with the Z disc. Now, in the middle portion of the cell, you have what we call as a thick filament, and the thick filament is made up of multiple myosin 2. So each of these dual heads that you are seeing is a single myosin head, myosin 2 molecule, another myosin 2, another myosin 2. And the way it is arranged, let's take a look at, so this is how it is. So you can see that a single myosin 2 will look like this. So you can see two heads and the tail. So the tail portion, the rod portion is at the middle, right here, is in the middle. And these are the club shape or the head. And see that this are the two myosin. So that if you just correlate with this picture right here, your Z disc is right here, is one Z disc, your another Z disc is right here. So now you have some names in here. So if you see that the zone where you have only actin next to the Z disc, one here, right here, one here, right here. And this area, and this is the next Z disc. Remember that this is the next sarcomere, this is the next sarcomere. This area where you have only actin and there is no overlapping myosin, we call this as an I band, right here, I band. And it will be present on both sides of the Z disc in a resting sarcomere. And then what happens, that if you actually see the area which is in the area where you have the actin and the myosin, they are overlapping, we call this as an AI zone. And if you just look at the first myosin head to the last one from each direction, the entire length of the thick filament of the myosin is the A band. So very interestingly, what we are going to see, the length of the myosin will never change, and the A band length never changes. It will always remain the same. A band, even in a contraction or relaxation, A band will remain the same. But what happens, the I band is the area where you have only actin and no myosin. When we have the contraction, both the Z disc come closer by. So if you just look at this structure right here, what's going to happen, the Z disc is going to move in this direction, because these myosin heads will going to pull the actins, and both the Z disc will move towards this. And you are going to see that this I band right here is going to disappear. This entire zone is going to be the AI zone, and the A band will be the length of this entire part. And this H zone, which is just the rod portion of in the middle, rod portion of the thick filament, this will basically disappear. These actins will connect to each other. So basically, just think about that. This is like you take your fingers, separate, and then you just do that. So when the muscle is relaxed, they are like this. And when you're contracting, you are just overlapping them. They're coming together. It's just exactly the same concept. So take a look at some of the supplementary videos, like the YouTube videos. It's very, very clear, and you will be able to see that. So let's take a look at a little bit bigger. And remember that this entire structure is absolutely stable. There is no polymerization or depolymerization of this actin happens in the muscle cell. Why? So the first capping proteins, see that this is the minus end. They are capped with a tropomodulin. There is no dissociation from the minus end. The plus end, we already saw that. They are capped by the cap Z. There is no dissociation from the minus end. No association at the plus end as well. No, so none of the end associate or dissociate. They are in constant shape. On top of that, if you just look at that, on the side, there is a protein called nebulin. Nebulin also stabilizes this filamentous actin from the side. So that's another protein. So nebulin. Now, the actins are associated with a Z disc, but the myosin looks like that they are floating in the middle, but it cannot be like that. It has to be associated with something. So there is a protein called titin right here, which is this coil-like structure, spring-like structure, that basically keeps the myosin associated with a Z disc. And when the contraction happens, that this will squeeze and then it will relax. It will squeeze, it will relax. So we are going to see that in a second. So here, see what has happened. This is a relaxed condition of the sarcomere. You have a Z disc, you have a Z disc, and here the myosin heads are interacting with the actin. See, right here is the I band. A band is the length of the entire myosin. And here is the I band, only actin. Now you have an ATP, calcium, everything comes in. The two Z discs will move towards each other. So they are moving towards each other. And see that overlapping actually increases. See right here, you have this I band and it almost shrinks to none. Shrinks to none. And ultimately what will happen that this Z disc will come all the way in here. So the I band will completely disappear and the A band will basically from end to end. And these will essentially touch. See that there are lots of gap and this was the H zone and this H zone will also disappear. This is the contracted condition of the muscle. And then what will happen that when it's relaxed, again, these titin molecules are going to pull them back. These are with the association of ATP. They will release the head and the Z disc will go back to its original position, which is relaxed phase of the actins. All right, so now let's take a look at how this whole thing actually happens. So there are two important ones, and definitely know the names of these proteins and the physiology of this, how it happens. So the bigger story, I'm just giving you a very small nutshell. We're not going into the detailed physiology of that, but just keep it back up in your mind that the entire story, that when a skeletal muscle is contracting, you get a nerve signal. There is a neurotransmitter that is released in a neuromuscular junction. And that neuromuscular junction actually creates an action potential or voltage across the plasma membrane of the muscle cell. And that voltage is transmitted into the endoplasmic reticulum of the muscle cell. And the endoplasmic reticulum of the muscle cell is known as the sarcoplasmic reticulum. Sarcoplasmic reticulum. This is, again, these are not needed. This is just reticulum. This is basically nothing but muscle ER, endoplasmic reticulum. And why this is important, and sarco is essentially sarco or myo. These are the adjective that we used with the muscle cell. So sarcoplasmic reticulum is a muscle endoplasmic reticulum. They store the calcium, Ca2+. This is the storehouse for the calcium. Calcium is a signaling molecule. It cannot be in the cytoplasm. It must be stored somewhere. Now, when this neuromuscular junction fires the signal, this voltage comes in. There are some deep crypts in here where the voltage is actually transferred onto the muscle ER from here. And so what happened, the sarcoplasmic reticular membrane or the ER membrane actually get these action potential or the voltage. And at that time, there are some voltage gated channels. They open and all these calcium actually flood the entire cell, the cytoplasm of the muscle cell. This is absolutely necessary for the muscle movement. So what happens in a normal condition, this is your filamentous actin associated with the Z disc in there. And on top of that, you have this band-like structure, which is tropomyosin. And along with the tropomyosin, there is another protein, which is troponin, is associated with that. So what it does when the muscle is in a relaxed state and there is no nervous intervention to contract that muscle, these tropomyosin actually cover the binding site of the myosin. See that these red things are the binding site of the myosin. In a normal condition, without the calcium, the tropomyosin actually covers these myosin binding site. Now what happens? Due to the neuromuscular excitation, the voltage comes into the sarcoplasmic reticulum. It floods the cell with the calcium. This calcium comes out and it binds to the troponin. See that right here, the troponin actually binds to the calcium, and the troponin changes the conformation. There are a couple of different TNT, TNI, and TNC, this calcium binding domain. These are, again, that detail is not necessary, but all you have to know is from there, the troponin binds to the calcium, changes the conformation, and lifts up the tropomyosin. It essentially lifts up the tropomyosin and it exposes the myosin binding sites in the presence of calcium. So now these red dots, these are the myosin binding site, they're exposed. And now the cross-bridge cycle can go on. There is ATP, and you have absolutely no problem of interacting. The muscle can relax. So after this whole thing is done and the nervous stimulation is gone, these calciums are going to go back by pump, like there are calcium pumps, which will pump all those calcium and they will go back to the sarcoplasmic reticulum. So what happens that you can just be able to correlate with that. When we try to do a workout or a strenuous workout, we first warm up. We do not stretch our muscle. We always warm up. So why the science behind that is that when you gradually warm up, all these calciums are gradually getting released from the sarcoplasmic reticulum, and they are ready for your muscle to work with. So your muscle will work at its maximum efficiency. Now, what happens that once these calciums are out, if you are just constantly doing that, these calciums do not go back. So it takes a little bit of time for the calcium to go back. So if you're just stimulating, so when you're running a 50-meter or 100-meter dash or playing basketball, very high-intensity sports, in between the movement, these calciums do not go back to the sarcoplasmic reticulum. They remain in the cell, and you can go back-to-back rapid movement of relaxation construction as it's needed for the movement. But at the initial stage, you need to give them some time for the voltage to activate these calcium channels to come in so that all your actin binding sites are actually open for that. So this is the process. So know the names of these proteins. That is the important part. Troponin is the calcium binding protein associated with the tropomyosin. Upon calcium binding, it lifts up the tropomyosin to expose the myosin binding site, which can essentially, with the presence of calcium, can lead to the cross-bridge. So only when this is open, at that time, only you can have the cross-bridge cycle, which you talked about right here. This cross-bridge cycle can only happen when the tropomyosin is lifting up and the myosin head can bind to the active site of the actin. So that's basically the correlation. All right, so we already saw that role of myosin 2 in non-muscle cell. What we can do, that these are some of the experiments that multinucleate cells are observed in the absence of functional defect of myosin 2, because cell division contractile ring cannot form. So you can just basically delete myosin 2 gene or inject an anti-myosin 2 antibodies. This can actually happen. Many other adhesion belt, stress fiber, cell-to-cell contact. These are all questions. So we just can ask some of these critical thinking questions. You have actually seen a cell which is multinucleated, and what could be the cause of it? Or you have deleted the myosin 2 in some of the cell, what you are going to see in those type of questions we can ask. Very straightforward. Let's start with the intermediate filament. This is a small one, but each one of them could be answer choices. So that's why they are in there. That just remember that which of the following are properties of that withstand mechanical stress, not dynamic like actin, do not shrink, grow actively. Along with that, remember that they do not have plus or minus end. Oh no, they are already here. Display similar cellular distribution as microtubules, radiate from the nucleus outward. They are not directional, no plus or minus end. The filament assembly does not require ATP or GTP. They are much more permanent structure in the cell. They do not have this dynamic nature that they dissociate or associate. So much more, not dynamic like actin. So each one of them, very careful look at that, because all of this will be a couple of different questions and answers, because mainly the way the questions will be asked is which of the following is a property or not a property of that. And when we are going to see that which of the following is a property for the intermediate filament, you're going to see that distractors will be essentially the property of the microfilaments and the microtubules. And your correct answer will be one of these. So just remember, so just giving you an example, that will say that all the intermediate filaments have a plus end and a minus end. So the intermediate filaments, when associated with an ATP, they assemble, or associated with GTP, they assemble. When they are associated with ADP or GDP, they dissociate. So basically, those are facts from the microtubules or from the microfilaments. Those will be the distractors. So just make sure that very carefully read those questions. Choose the ones which is absolutely specific for the intermediate filaments and not microfilaments. Or one thing very specific in here, right here, this is the nuclear lamina, or underneath the nuclear membrane. And there is a specific protein associated with that, underline that. And if you look at that, this is called lamin. So nucleus, nuclear structure organization, and here is the distribution. So that is underneath the nuclear membrane, and we call this as a nuclear lamina is the structure, and the lamin is the protein of the intermediate filament. Now, if I go back all the way, the first picture that I talked about, those right here, these green proteins, these are not lamin, but these are laminin. MININ. They are two different proteins. I'm not going to ask about this, but just make sure that these green structure are laminin, NIN, versus lamin in nuclear lamina. So they are two different things. By chance, if you're reading the textbook, don't get confused on that as well. Entirely different distribution. And both are lamin, laminin, both are intermediate filaments. All right, so here we are looking at a typical structure. You have a monomer, then you have a parallel dimer, and then two of these parallel dimers, two of them come together and form an anti-parallel tetramer. So see that you have N-terminal end, C-terminal end, two of them, and now you have the C-terminal end on this side of parallel dimer and the N-terminal. So they are anti-parallel. And two of these anti-parallel structures form a protofilament. And you have, this is one. So you have 1, 2, 3, 4, 5, 6, 7, 8 protofilament form a protofibril. And then you have 1, 2, 3, 4 protofibril form an intermediate filament. So again, out of those, it may sound complicated, but most importantly, just remember that because this is the unique part, that's why just remember this part, not all of them, but this will be in front of you, that this is anti-parallel. So two parallel dimers actually form the anti-parallel tetramer, and that forms a protofilament of the intermediate filament. So that is the important part. So definitely remember that anti-parallel, C-terminal on one side, N-terminal on this side. I will just change that color. A lot of people actually miss that question. N-terminal on this side, C-terminal on this side. And that is what your anti-parallel essentially means. Just an EM. Is two anti-parallel tetramers forming a protofilament, or anti-parallel is? So here is one protein. So when you are forming the dimer, you have two of them forming a dimer. Now, when you are having a tetramer, you have two of these here, two of these here. And when you are taking a protofilament, you are taking this whole anti-parallel tetramer and you are putting two of them here, two of them here. So now you have two of the protofilament, and each of the four protofibril, in each of them you have two protofilaments. So essentially, I'll just draw it in here. If you just look at the protofilament right here, you will have, this is 1 and 2. So this will be considered as 1 here. So you will have two of the protofilament here, two protofilament here, two of them here, two of them here. So you have 1, and then you have 1 plus 1 together. And then you have these 1, and you basically, so in here, so this is essentially what you are seeing. So 1, then 1, 2, then 1, 2, 3, 4. And then as you do not have the C and N, it becomes a little bit confusing in here. But just remember that each one of them will have 2. And then you have this. Here you will have 2, 2, 2, and 2. So this entire protofibril, that entire bundle will form one of these, one of these, one of these. So that's how it goes. So just make sure that this will be in front of you. So definitely take a look at this when you are answering that question. So definitely remember that. All right. So here, what we are looking at, a very straightforward type of question, but a little tricky. Sometimes if you just see that these are all the different classes of those, and the important part is that all of these proteins of the intermediate filaments are different. See that you have one class of these intermediate filament which are made of acidic keramines, which acidic means that these do have a tendency to have acidic amino acids a little bit more. You have more of aspartic acid, glutamic acid. They are rich in those amino acids. Whereas if you have a basic keratin, you have more of asparagine, you have more of glutamine. Those type of basic amino acids are more in these basic keratins. So acidic keratin, basic keramine has nothing to do with pH or anything like that. It is the composition, that whether they have more acidic amino acid versus more basic amino acid, just depending upon those. Then you have a desmin, then GFAP. These are glial cells associated proteins, and then they are different cell. Then vimentin, neurofilaments, and LMM means like these are large or long, medium, and then high. These are the different ones. Lamine, you can see. So basically, the take home is, remember that, as I said again, all of these are different proteins and forming the intermediate filaments of different classes in different cells. And out of those, I would just highly suggest that definitely remember this one is the lamine in the nuclear lamina. Nuclear lamina. That is, see that right here, this word nuclear lamina is not here, which is this essentially, that blue structure. That's why people actually miss that question. So I'm going to show you that where you see that nuclear lamina so that you can connect that. Right here is your nuclear lamina, right here. So connect that nuclear, there is always something in here. So this is nuclear. So just remember that. This is nuclear lamina. This is essentially the intermediate filaments underneath the nuclear membrane, which is a double membrane. These keeps the integrity of that, and that is made by the lamine. If you remember that, you should be absolutely fine. Another very important thing, that you have motor proteins associated with your microfilaments and microtubules. You have your myosins, and then you have a kinesin and dyneins for your microtubules. There is no associated motor proteins with the intermediate filament. That's also a very, very big difference between those. So mainly, most of these, if you just look at that structural organization and tissue integrity, and those are essentially the function. So basically, rigidity is one of the main function of the microtubules. All right, so this will do it. Neurofilament, glial filament, these are all good. These are your keratins, which are the abrasion-resistant protein that if you have a very dry skin and you scratch and you just get those ashy structures, those are the top layer of the dead cells of your epithelium, which are filled with these filamentous keratin. And those are essentially your acidic and basic keratin that you have. And even when your cell's top 20, 30 layer of your skins are dead, the entire cell is filled with the keratin, which gives you that abrasion resistant as well. They are 200, 300 layer thick in your palm and the sole of your foot, but at least 30, 50 layers on your regular skin as well. They're all dead, but filled with these intermediate filaments. That will do it. All right, last part that we are going to do the microtubules, basic structure. These are good. Yeah. So very important slide. A lot of questions can be asked from there, several one of them. So the first thing is that they are very similar in the sense with the microfilament because they have a plus end and a minus end. And the subunits do the treadmilling just like the actin monomers. So that's important. But the difference is that the actin is ATP bound and hydrolyzed to ADP bound, and it moves. Whereas in here, you have two different subunits. You have an alpha tubulin bound with a GTP, and then you have a beta tubulin, which is bound with a GTP, but as it moves towards the minus end, that GTP is hydrolyzed into the GDP. The GTP of the alpha tubulin is completely structural and never hydrolyzed. It is always GTP bound, always. This is never hydrolyzed. Only the beta tubulin hydrolyzes its GTP to the GDP. Here it is just shown as GDP, but remember that when it's added towards the end, it is GTP bound. So remember that. So if you look at the structure, what happens at the plus end, the beta tubulin is at the plus end, and the lighter ones is the alpha. So the alpha is not facing the plus end. Alpha is added first and then beta. So alpha, beta, and it goes. And the minus end, that is the alpha tubulin end. See that lighter alpha tubulin as the minus end. So remember, alpha tubulin end is which end? Beta tubulin end is which end? Now, there is a gamma tubulin, which has nothing to do with the final structure of the microtubule, but this is the nucleation factor. So if you just remember that you have the NPF or nucleation promoting factor for your actin, the same way the gamma tubulin is essentially a scaffold on which the alpha beta tubulin start polymerizing, and gamma tubule goes out and the filament is formed. So if you look at a single filament right here, you have a protofilament, and multiple of these protofilament come together and form a hollow structure, which is a microtubule about 25 nanometer wide. So again, alpha beta tubulin dimer, beta tubulin exchangeable GTP site, which hydrolyzes a GDP. So again, the way it is that when it is the beta tubulin is GTP bound, so it is GTP plus GTP, then it has a high affinity for binding. As it treadmills, that GTP of the beta tubulin is hydrolyzed to GDP, and at that time, it will tend to fall off. But again, remember that natural hydrolysis and low affinity is not sufficient for the speed that they need. So you need some of the dissociation protein, which will take off these subunits in a much faster rate, just proteins similar to the coffilin or the gelsolin for your actin. So we are going to see that. So all of these things are very important. So know in every detail, very, very important. The next is also very important because you have to connect that with another slide. All right. So microtubule structure, what you are seeing, this is where you have 13 protofilaments. So what we mean by this is right here. If you just count, you have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, we'll have 13 of them. So each one of these protofilament is just one. So here you have a singlet where you have a 13 protofilament, sometimes 11 to 15, but this is the most common one. When you find these microtubules across the cytoplasm, that the most commonly we see throughout the cell, say for an example, during the cell division, the chromosomes that move on these are all singlet microtubules. They are all these 13. Whereas you find these doublets, which is a 13 plus 10, because these are shared with both, you find them in the cilia and the flagella. And this is the one a lot of people actually miss. There are some places where you see this 13 plus 10 plus 10. You find these in the basal bodies, which is at the base of the cilia flagella, and the centrioles, which is nothing but the mTOC. So where is a subtle difference? And just make sure that you get that one. And I'm going to connect another picture with that. So if I just draw a cilia or flagella like this right here, you are going to see these doublets right here like this all throughout. Here you will have the doublet. At the base right here, you will have the basal bodies right here. That is this. And this structure, which is the entire length of the cilia flagella, they will be like this, which is a doublet. This is the basal body. Remember that. That is triplet. So the ring-like structure, and you see that in the centriole as well. All cilia flagella basal bodies have triplet microtubules. So let's see that where we see this picture in a proper way. This is the one that I see a lot of people actually miss. So I will go to the very last picture. So here, if you just carefully look at that, cilia or flagella, you have 13 plus 10. And here you have a singlet, but it's a doublet in the middle, but these are 13 plus 10. We are talking about this. This is the body. So this is this entire body. But where it starts from the cell, which is, see right here, these are 13 plus 10 doublet, just missing that. But this is the picture. So this is the picture where you are seeing this structure. And if you carefully look at that, you have 2 dots in here, 2, 2, 2. So these are your 13 plus 10. Right here is the basal body, which also comes to the mother and the daughter centriole as well. And if you carefully look at that, you have 1, 2, 3 dots, 1, 2, 3 dots, 1, 2, 3 dots. These are the basal body with the triplet microtubules. Here it's doublet. These basal bodies will have the triplet. So that's where this is a very common question that is actually missed by that. Where are the locations for this? So definitely connect these two things, which is here. So just remember that. All right. So microtubule radiate from the mTOQ, essential for the cell division, and stable, long-lived, remain polymerized for a long time in your axons as well. They, with the help of the motor proteins, they move the neurotransmitters and very long distance. And remember that the microtubules are the one on which all the neurotransmitters are carried in vesicles from the cell body towards the periphery of these telodendria, which are at the end of the axons from where the neurotransmitters are released. You see these intermediate filaments. The intermediate filaments is essentially for the structural integrity of these long axons of the nerve cells, total structural integrity, so that when you move, they don't tear apart. But all the movement of material happens on these. So where are these movements? Remember that all these neurotransmitters, many of these are protein molecules, are synthesized through the secretory pathway, through the ER, through the Golgi, loaded onto the vesicles. And then these motor proteins move them from there all the way to this end. And that's what we are going to essentially see. So again, mTOQ, any structure used by the cells to nucleate and organize microtubules. Of course, you have the centriole in the middle of the cell if it is a dividing cell, but the basal bodies are also known as the microtubule organization center, and they could be anywhere. So mTOQs can be separated with the dendrites and axon of the nerve cells. So definitely remember those as well. Very straightforward question. Now let's take a look at these. So here is an interphase of the animal cell, which is in a resting phase. So what you are seeing that this is mTOQ, which is the centrioles, which make the microtubule, and you have 2 of them. And the orientation in here in a normal cell, the minus end, which is an alpha tubulin end, is towards the mTOQ side. Whereas towards the cell periphery, that is your plus, towards the cell periphery. So it grows from the hair towards the cell periphery. The same way here you have a flagella or cilium. Here you have the basal body. That is also an mTOQ. That is the minus end. This is the plus end. So it grows towards the plus end. In a normal cell, this is true. So always the plus is towards the cell periphery. And see that what is happening right here. You have the mitosis, and in the mitosis, there are proteins. From the minus end, it actually shrinks, as well as the plus end with other proteins, it shrinks and it pulls the chromosome towards the pole. So these are the orientation and helps in different processes, and we're going to learn that about those. Nerve cell does have a very unique structure, and that is basically the question. In the axon, you can see that you have the minus ends towards the cell, plus end in here. But dendrites, sometimes this orientation can be entirely different. So your minus could be towards the periphery of the cell, and the plus end is towards the cell body. So we just do ask that what it is. And this is for the motor protein movement, because there are two sets of motor protein. Your kinesins move from the minus end towards the plus end. Your dynein moves from your plus end to the minus end. And in the dendrite, this is the receiving end of a nerve cell. So everything moves from the periphery towards this and back and forth, different movement. Whereas what you see that in the axon, most of the things that are moving through the axon goes from the cell body towards this. So these are usually done by the kinesin. But these can be back and forth, and to aid the process of kinesin and dynein, both can take part of this. So that's an exception. Know that. This is your treadmilling, and we are not going into the critical concentration. Is the same way, just like your actin, you have a critical concentration at this end, critical concentration at this end. If this is lower than the critical concentration, then it will be added. If it's higher than critical concentration, it will be dissociated. And you can see the treadmilling, exactly the same thing, not important. A couple of different things that is important here, different reagents. If you actually cool a cell, it actually stops cell division as such. And how you stop the cell division? Because it will not form the spindle fiber to divide the cells. So if you just warm that, again, microtubules will go. If the cell is wants to divide, it can actually divide. Or you can add a chemical which is known as a calcimide. And sometimes if you just see a word colchicine, they are similar. One is from a natural product, one is an artificial. This calcimide also disrupt the microtubules as well. So we are going to see the effect of those in here. So in a second. So here you are seeing this colchicine. Remember that this colchicine and here calcimide, they are the same, just a little bit of different in the structure. So what happens? They bind between the alpha beta tubulin on the dimers, and this is the important one. That is a lot. It's in between lines, but it's just in front of you. Don't miss those. So again, I'm underlining. High concentration depolymerize. Low concentration actually stabilize. So very different function of the colchicine and calcimide. Both of calcimide, they are the same thing. But again, I will specifically ask for that. On the other hand, if you just look at the Taxol, they actually stabilize. And the Taxol, anti-cancer drug, essentially. So what happens, just think about that. You have a cell division, and these are your mTOQs. You have the spindle fibers right here. Your chromosomes are aligned in here. You need to shrink the microtubules so the chromosomes are pulled towards the pole during the anaphase. And if you stabilize the microtubules, they are in this condition and they cannot move, and that will arrest the cell division. If you remember, in the very first chapter, we actually talked about different drugs that are used, and one of them was monoastrol. Again, if you remember, we're not going to ask in here, but monoastrol, which on the other hand actually depolymerize the same way the microtubules where the chromosome cannot line up, they cannot go in there. So many of the anti-cancer drugs, which actually arrest cell division, are either stabilizes the microtubules or they disrupt the microtubules. Both of them will be detrimental for cell division. All right, here we are looking at some of the, as I said, very important. We are just going to ask the similarities with that, both plus and, but this is important, nucleotide, ATP, GTP, and rest is all the same way. Just remember that only difference is that microfilaments, you just only have the actin, and in the microtubules, you have the alpha tubulin and beta tubulin, whereas the alpha tubulin GTP is bound permanently, beta tubulin GTP, GDP can be exchangeable. All right, so here you have some of the proteins associated with the microtubules. And if you just look at the cross, what you are essentially looking at, you are looking at a cross section of a cell. So if I just draw the cell like this and I cut it from here and I'm visualizing from this side. So I am looking at the cross section of that, and all these microtubules are running like this. And I am looking straight at each one of these as a microtubule. So here you can see that the microtubules are running and they are a little bit more spaced and these are a little bit dense. This is a nucleus right here. This is the nucleus right here. So they are a little bit dense. So how the cell actually can arrange that depending upon that. So there are some proteins which are called the microtubule associated proteins, and these proteins are known as the spacer proteins. So this is MAP2, which has a longer arm. So you can see that it has a longer arm. Whereas in here, there is a microtubule associated protein, which is a shorter arm. So what happens that if a microtubule has a longer arm, the next microtubule can only be here. Whereas in here, the next type microtubule can be here. So these microtubule and these microtubule are close by in here. That's what you're seeing. Here, they are a little bit more apart. That's you are seeing. So sometimes we can give you a picture. Say, for an example, what we do that we just give you a cross section of that, which is like this. And then you can see some real electron micrograph of some of these. And sometimes you will see that some of these pictures will show you microtubules like this. So this is the distance, this is the distance. But sometimes you will see some of them are like this, something like this. So you can easily see the distance in here is much less compared to these. So this is essentially looking like this. So if we just say that you are doing the experiment with tau and a MAP2 or a protein X and Y, this protein looks like this protein essentially look like this, which one does have a spacer arm. So in between the microtubules, if you see more space, it will have a more spacer arm like a MAP2. If they're closer, they will be more like a tau. So as we are using these exact examples, we usually just give you the example with MAP and tau that you have engineered these things. So just go back to this picture. And if you don't remember, just remember that your MAP2 does have a longer spacer arm compared to the tau, which has a shorter spacer arm. Just remember that, shorter spacer arm. So less compact versus compact. So these are the type of things that we essentially ask. All right, so these are some other proteins, and there's one phenomenon that happens, which we called at a catastrophe, and we will come to that. So here, what we are looking at, these are the tubulins, which in red, and here is one protein which is associated with here is an EB1. So here you can see that these are basically plus and binding proteins. And here there are EB3s. There are different proteins which are associated with those. And so EB1, EB2, EB3, many different proteins. But again, the important part is that what you are seeing at the end of the microtubules, at the plus end, there are some of these proteins which regulate the growth at the plus end or the shrinkage at the plus end. So what we're seeing that this is what is called a kymograph. And what we see that when you see these green structures, we see that in here you have a growth. So see that this solid line is a growth right here. And then at one point, the C, what we call this is actually called a catastrophe. And after that point, you can see right here, there is a shrinkage of the microtubules and there is no green proteins in here. So it's shrinking. Then R is a rescue. That means these plus and associated proteins again associate and it again grows and again shrinks, again grows, again shrinks. So this is a process by which these oscillations and association, dissociation of these plus and binding proteins grow that. This is happening in a very, very fast manner. So this is a property, or basically a catastrophe rescue is a property of microtubule growth, but we do not see this with actin. So this is very specific for microtubule association. So definitely remember this. All right, so now we are looking at what are the different proteins and how they basically regulate the disassembly. Two different proteins, they break the microtubules in two different ways. And this is the most interesting part. See that this is showing you that this is happening at the plus end. Remember that they do dissociate at the plus end as well as the minus end as it is required. So for example, these proteins will be working at the plus end, at the minus end. When this movement of the microtubules, chromosomal movement actually happens, this is super rapid. This happens with less than within 45 minutes from the metaphase to the anaphase. And the depolymerization happened at the plus end as well as the minus end. And the stathmin and kinesin 13, although it's shown here at the plus end, here it's not shown, but they do this in both ends. So here, basically what they are doing, the kinesin 13 is a motor protein that binds and bends these microtubules and break them apart. And this is an ATP-driven process. There's another protein, which is a stathmin, that enhances the GTP hydrolysis. Remember that when the beta tubulins GTP is hydrolyzed to GDP, it will fall off. And these are the proteins which regulate microtubule disassembly, both at plus and minus end. That's important. All right. So after that, we are looking at the motor protein, plus end kinesins, minus end dynein. Remember that there is always some differences in here. If you look at the dynein, cytosolic dynein, it goes to the minus end. Kinesins are plus. Again, you do not need to memorize any of them, but look at this kinesin NCD. This kinesin NCD that we talked about, that NCD20 just briefly, we'll talk about that later, nuclear cell division protein. This goes to the minus end. This kinesin, which are supposed to go to the plus end, this is an exception, just like your myosin 6. So myosin 6, remember that all myosins are plus end directed motor. The only myosin 6 goes towards the minus end, and this kinesin NCD is one of those. Dynein, this is minus end. Dynein is always, there is no exception in dynein. There could be, but I do not know top of my head, but we do not have any exception in the dynein so far. But kinesin, there is just one example of this. If you look at the kinesin, very similar to the structure, you have a globular head, then you have a coiled structure. The only difference, just remember that is usually a question here. You have a light chain, tail region, very specific question, tail region in kinesin. Remember that. This is in the tail region in the kinesin. These are essentially important with that. Whereas if you just look at very similar things earlier, all the way in here, right here, see that your light chains are in the neck region. So in the myosin, the light chains are in the neck region between the head and the stalk, whereas in the kinesin, they are associated in the tail region. Very specific question. Just remember those as well. Yeah, so that will do it. All right. So here we are looking at the different types of kinesin, just like you have myosin 1, 2, 5s, just like that. And organelle transport, they can, like, these are robust proteins. See that actin-related proteins like the myosin, they move small vesicles. But these actually rearrange organelle. That means that they can move around mitochondria and Golgi stacks. I mean, like chloroplast, they can move these large structure inside the cytoplasm. Very, very powerful motors. On the other hand, we are going to see that kinesin 5, they are bipolar, means that they have two poles. So see that these are the heads which actually move. But here you have this pole associated with one microtubule. This is associated with one microtubule. Both are plus end directed motor. So think about that. It is basically putting its foot on this plus end, and it is going towards the plus end. And if you think that these are its hand, it's pushing this top microtubule towards this. And this essentially we are going to see in the middle portion of the cell, which actually stretch the microtubules to increase the length of the cell during the cell division. We're going to see that kinesin 5. And see that right here, now the kinesin 13 actually shows in here that it can bind the plus and minus both ends for the end disassembly. That's also seen at the plus and minus end disassembly during the cell division as well. Very straightforward. So know all of these. Kinesin is plus end directed motor, contains heavy ether light chains, requires ATP and the cargo to different cellular destination, including the cell periphery. So here I did not put a star. The movement is a little bit different compared to the myosin. And that's why I kind of omitted that, because I think that people actually confuse that here. But just in a brief nutshell, I'm just still explaining that. See that it's a dual head, and this is the motor head, and you have a linker, and an ADP is associated when it is right here. So remember that when the myosin head is associated with the actin, globular actin, the ATP binding site is empty. Here it is an ADP bound. Now, here what happens, the leading edge binds to an ATP. Binding to an ATP to the leading edge swings the back arm forward. Binding to the ATP induces conformational change, causing the linker to swing forward. Very different. Binding happening here, but this is swinging forward. And new leading head finds a binding site of the microtubules, and then this PI is released. This is ADP, then ATP goes out. And now another ATP will bind, and the back arm will again swing forward. So it's just a little bit different process compared to that, but I will just not ask step by step. This is not required in detail. So structure of cytosolic dynein, and this is a very, very large protein and is an amazing structure. If you just want to see some of the discoveries of these tiny kinesin and the dynein, you can look at some of the supplementary videos. These are iBiology videos. And again, this is for completely personal interest, not required for your exam. And a lot of people actually ask is that what are the things that we need to know in here? So just I will say that know that proteins, first of all, large proteins, and they have a cyclic ring-like ATPase domain. See that? It's a hexamer, a cyclic ring-like ATPase domain. ATP hydrolysis move these rings to put tension on the linkers, which are associated with the cargo, moving them essentially forward. So take a look at I'll say that just fast forward and see some of the supplementary video that how it actually keeps the feet up, and then it will oscillate, plant it, and then move. It's really amazing that how at the molecular level, they actually found these things out in here. All right, so here you are looking at this whole structure, and this will be, again, you do not have to memorize that, but this is the whole thing, the cargo, and we call this entire structure, which contain actin. Now you have the ARP1 filament, capping protein, so even actin is associated with that. And then there is a protein called dynamidin, and this whole thing is known as a dynactin. So here I will specifically mention something specific. So remember that. If you remember that from your module 3, when we talked about the pinching of the vesicles, remember that your pinching of the vesicles, there was a protein which needed a GTP to pinch off vesicles. Name of the protein is dynamin. See that dynamin is the protein. Here it is dynamidin, dynactin, dynein. So very, very confusing in here. So what I will do that just keep up a piece of paper and then write it down that where these things are. So you have these three for dynactin, dynamidin, dynein. So dynamin is the help of the GTP. It pinches off those. So I will like at the end, once we've done that, I will just go back and show you that where you have this module 3 dynamin, because it is important. Because what the reason I'm showing, I will probably ask this and put all of these as distractors, and sometimes people put, or I'll just ask one of these and put those distractors. Sometimes people get confused with that. So dynein is the motor, remember that. And this P150 glued, this is a 150 kilodalton protein, and this is associated with this whole dynactin complex. This dynactin, individual protein names are not dissociated. And this is associated with an adapter and binds to the cargo. So this is how I'll say that you take home message from here. Just think about that. This is basically a car pulling a trailer. So just think about your dynein is the car. Your P150 glued is like, sometimes you have seen that these big trailers which are carrying those trailer homes in there. There will be a car at the back and car at the front. This P150 glued is basically keeping this entire cargo integrity in there so that they don't fall off. So that's basically the function of that. Then this is the hitch and the coupler, and then you have the cargo. So if you just think about that, that's what it is. This entire complex is known as the dynactin complex, and there is a specific protein right here called dynamycin. And there is a story for that why they name these things in such a weird way, that when the scientists actually were working on this, how they reconstitute these things, they make all these recombinant proteins, reconstitute them, and then try to see these movement by single molecule fluorescence microscopy in vitro or in single cellular condition. And they have seen that if they overexpress or produce too much of this dynamycin in the cell than the molar amount required, that entire thing falls apart, blows apart the entire structure like a bomb. And that's why they named that protein as dynamycin. So if you just produce, so just the right amount is needed. If you overexpress the dynamycin, it doesn't form this complex essentially. So sometimes these are some small things that actually goes into your long memory. So remember that. So dynactin is the big complex which associate with the adapter and the cargo with the dynein. So this is what is different, and differentiate this with the dynamine, which we are going to see that. Your ATP bound or ATP not bound movement, as move that, again, detail is not necessary. This is showing that how the different motor proteins are moving different structures. So dyneins are moving, see that from minus end, so plus end to the minus end. This is kinesin. This is moving from towards the plus end. So if you just look at that, that is happening. Now the interesting thing, what happens, suppose this kinesin is moving and going to the plus end. How does it actually come back to take another cargo from the minus end? Because it is stuck. It cannot move. If it reaches this right here, it cannot go back towards the minus end, because that is dynein can do that, it cannot. So a very interesting phenomenon is observed that is called the hitchhiking phenomenon. So what happens, see right here, there is a dynein which is moving towards the minus end. This is a granule or a cargo where a kinesin is associated with that, and that is not associated with a microtubule. It's just hitchhiking with that, and it will go back in here. So right here, on the other hand, you can see that right here, a dynein is essentially hitchhiking and moving with the kinesin. So if dynein end up in here, how it goes back there to come back again. So as the kinesin is moving towards the plus end, it is hitchhiking with that. So it gives you an idea that how the movement happens. Now, think about that a microtubule, these are your freeways or the superhighways. On this, the dynein and the kinesin actually move the cargo at a super rapid speed. And then they need also a precise delivery. And from the microtubule, they transfer their cargo to an actin-based delivery system, which is much more intricate, and myosin can take over the cargo and deliver. Think about that from a manufacturing facility, a big semi loads up all the material, and then a small delivery truck actually goes door to door to distribute that material. So amazing process. And you can take a look at some of those supplementary videos. All right. So after that, we are talking about a particular dynein, which we call as an axonal dynein. And they are associated with the flagella and the cilia. And for animal cells, the only example that you essentially see in the sperm or the single cell eukarya, some of the protists, you actually see these flagellated movements. And the cilia, many per cell, locomotion of single cell eukaryotes like Paramecium and those kind of things, you see those cilia. But cilia, there are lots of different areas. We have our uterine tubes have ciliated cells. Then we have our trachea. They are lined by these ciliated cells, which actually move material constantly, moving particles or fluid over cell surface, tracheal cells. We see these everywhere. So how this is associated? So if I just take a cilia or flagella and make cross section, what we see that you have a 13 plus 10 arrangement of 9 of these. So you have a 9 doublet and central 2. So we call this as a 9 plus 2 arrangement. And you have an A ring with 13 and the B ring with 10. And now, these are the ones which you are seeing, the green ones. These are your axonal dyneins. So you can see the dynein. These are the outer and the inner dyneins, outer arm, and these are your inner arm. And all of these doublets are connected with a linker protein, which is known as nexin. And now, interestingly, what happens that you can see that we call this as the A tubule and the B tubule doublet. And these heads of the dynein is going to bind to the next one and pull them towards them. Then these are going to bind them towards them and pull them towards them. And this movement actually goes from one of these doublet across that entire part. So think about that. When you are contracting this part of the structure of the flagella is bent, this is relaxed. Then the movement actually goes on the other side, which is bent. And this is how this corkscrew motion actually propagate through the entire part. And that gives you that undulating movement. So it is just constantly happening like this. It's going from one end to the other. So that's essentially the structure. So this is the base and these are the heads. So these are the heads and this is the base. So let's take a look. So here you have the adjacent B tubule, you have the minus end, plus end. And here is a stable association with the A tubule. This is attached, and think about that. They are kind of hanging in there. And dynein is a minus end directed motor. By the help of ATP hydrolysis, they're moving towards that. Now, the structure is very different from the dyneins, which actually move the cargo. So structurally different, but they are the same kind of protein. So this actually gives you an idea. So if you think about that, this is your A and the B tubule, although this tiny drawing from this is actually wrong. If you just look at that right here, this is your A tubule with 13 and B tubule with 10. This is correct, whereas they kind of mess this up where they make the A tubule 10 and the B tubule 13. So basically the 13 will be should have been the A tubule, and this would, but again, it doesn't take the merit away from what you need to understand from there. So what you are seeing, dyneins are associated all throughout the length of these doublets at the base right here. And these are the heads associated with the A tubule of the next one. So remember that. So what happens that when you have the activation, the head of the dynein trying to move towards the end of this, towards the minus end. So from here, they're trying to move forward. They're trying to move forward. And they are all in a single sheath. They are connected with these nexin molecules. These are the nexin linkers. So the only way that if you just pull this and this is not going anywhere, the only way this can happen is that bending of this. And this causes the bending. And this is happening in just 2 of these sets right here. Bending happening to this, then this will happen to this. And as I said, that it just goes around from one end to the other towards the entire length of the cilia or the flagella, which causes the movement. So how it works like that and what's the role of the nexin? The scientists actually remove these nexin linkers by the proteases. These are very specific engineered nexin, which does have very specific protease digesting sites, which basically only remove nexin, doesn't touch any other proteins. And here, the AB microtubules, the doublets doesn't have the cross-link nexin in here. So basically what happens in here, these pink things are not there. So they are all individual, but they are not connected. And now you just add the ATPs and activation of the dynein. What happens, these AB tubules slide past each other and they do not bend. So only due to the nexin, they can have this undulating movement and they do not bend. Remember that head of the dynein associated with the B tubule, when the head of the dynein trying to move towards the end of the B tubule, the bending of the tubes, microtubules cause the movement. All right, so a lot of highlighting here, which is what we call as an IFT or interflagellar transport. So if we're just looking at the flagella, this is the plus end towards the periphery of the cell. This is the base towards the middle portion of the cell. So there are two types, which we call as an anterograde movement and retrograde movement. As you can see that if it's moving from the cell towards the periphery, we call this as an anterograde movement. If it is coming towards the cell, that's a retrograde movement. So any retrograde, anterograde movement, kinesins are essentially going to move the material, and the dyneins are hitchhiking. Whereas if you see a retrograde movement, you can see that dyneins are carrying the cargo. Think about this way, that there are some material that are needed. These are signaling molecules and other stuff. They are needed at the tip of the flagella. And the tip of the flagella actually receive some of the signaling molecules from the environment. They needed to be brought back towards the body. So these are delivered here. Then they change. They see that they are switching from the kinesin to the dynein, and they are actually bringing these material in here. So again, can travel to the tip of the cilia flagella as they have the microtubule plus end. In order to travel to the tip, the hitchhike cargo carried by the kinesin 2. And in absence of kinesin 2, dynein will not be able to reach at the tip of the flagella, and retrograde IFT will cease. So it's a typical question that we ask, that if you actually mutate a cell where you do not have kinesin 2, so what will happen? If you miss any one of them, the protein that needs to go back, they can't. So essentially, you do not have the kinesin 2, okay? So what is happening? All these dyneins are moving the cargo towards the body of the cell, and they are stuck there, and they cannot go back to the plus end. So the retrograde movement will essentially cease. They cannot bring anything from the plus towards the minus end. But see that what is happening. So know the idea. Kinesin 2, once they reach the tip of the flagella, they go backwards, the base of the flagella with the minus end of the microtubule, hitchhike on the cargo carried with a dynein. If we do not express the dynein, kinesin 2 will accumulate at the tip of the flagella. These blue things will be all in here. They cannot come back because we don't have the dynein. So anterograde IFT will cease. See that if they are in here, for the anterograde movement, you need the kinesin to be brought from here. These needed to be brought by hitchhiking in here. So if you do not have the dynein, anterograde movement will cease. If you do not have the kinesin, you have the retrograde movement will cease. Although they by themselves going in the opposite direction, but it helps the other motor protein to go back. So that is the rationale. That's why I just did that. So just make sure that you have this slide when you answer that question, that what is exactly happening. It's the opposite one. Primary cilia, responsiveness, these are all very straightforward. And the only difference what you have, that they are non-motile. They can move with the flow of fluid like urine or anything like that. They can sense the bending or the pressure of the urine and all those kind of things, but they do not have the ability to move, and they do not have the central doublet, the cilia, like motile cilia and flagella. So very different. These are your motile cilia flagella, 13 plus 10 arrangement in 9 plus 2. These are just 9 doublets in the outer part and do not have the ability to move. These are some of the ciliopathies, some of the disease that actually happen when you have these problems in here. So these are some of the mutation that happens with this PKD1. This is called a polycystic kidney disease, PKD1 and PKD2. So what happens that these actually sense the urine flow, and the PKD1/2s needed to be delivered from the cell by the cargo anterograde movement to the cilia and the flagella. And if they have a mutation in there, they are not delivered onto the primary cilia, and they cannot sense that, and that can cause the problem of the urine production, and polycystic kidney disease can actually happen out of that. So here is another kind of ciliopathy. It's called the Bardet-Biedl syndrome, polydactyly. These are all straightforward. And here is very interesting. This is where you have one of the 14 genes encoding the BBsome. Now, BBsome is a very similar structure like the COPS or the clathrin, where it actually forms the cargo which is associated with the membrane. So that is a BBsome. So the cells have normal distribution of kinesin 2 and dynein, but they can't associate it with the cargo in the absence of the BBsome. So just think about this. That will just make you in perspective. So you have the cargo, you have the dynein, and here you have the different proteins. And somehow, if you just miss this ARP1 or any protein which is in the dynactin, then this cargo and the dynein association will not happen. And BBsome is one of the protein associated in the membrane. It's not like the cargo, it's a cargo-related, membrane-related. And that's the reason the cargo cannot be associated with the motor proteins, and they are not delivered at the tip of the flagella, and that causes the problem. So that's sensory receptors can be trafficked by the saliva to the transport. So this actually gives you all the events in the cilia flagella. You can see that right here. These are interflagellar transport cargo complexes, and you have a kinesin and the dynein. So see that on the microtubule here, the green one is the kinesin 2, that is moving towards the plus anterograde movement, and dynein, the purple one, is hitchhiking. It reaches the top. See that it delivers the cargo here. Now it is bringing cargo from the retrograde transport. See that now it is switched. The purple one is the dynein that is moving the cargo, and the green one is basically hitchhiking. So that's essentially the process. Also know that this is your 13 plus 10, 9 outer tubule and 2 central tubules, because this is a flagella or cilia that is motile, and the basal body do contain 3 of these 13 plus 10 plus 10, triplet of these, but they don't have the central doublet. Basal body or the centriole doesn't have the central doublet that is only present in here. So just remember those things in here. All right, I think that essentially will do it. So any of you guys, if you have a question, send me. And if you need any one-to-ones, this is for everyone because it's being recorded. If you need any office hours or anything like that, just feel free, and I will be more than happy to do that. So this is the one which is the toughie. If you just go over this, then you will be in a very comfortable zone. So you will, with the third and the fourth one, you will be all smiles. I guarantee that. So this one is the most challenging ones that if you just go over this hump, you should be all good. Good luck with the exam. Best. See you then. Bye-bye.